Today we're going to speak about the origin of life. How likely was spontaneous generation of life? We're going to have a brief review. Which one is real science? The data must conform to the theory. If it does not, adjust or ignore the data. Or the theory must conform to the data. If it does not, adjust or abandon the theory. Science is supposed to be observable, testable, repeatable, and falsifiable. Most creationists are honest enough to admit that creation is not observable. It happened in the distant past. It's not testable. We mortals certainly can't repeat it, but it is falsifiable, meaning we can check it against known data. If evolutionists were equally as honest, they would also admit that evolution is not observable. It also happened in the distant past or is happening so slowly that it can't be discerned. It's not really testable. It's not repeatable, but we can falsify it against known data. Also, an assertion we've made from the beginning, both evolution and creation are religious beliefs. But which one fits the data best? And that's what we examine in all of these videos. I'd like to introduce a new term to you. It is the term simplistic. Simplistic is not just a fancy word for simple. Simplistic does not describe things that are easy to understand. Those things are truly simple. However, if something is misleadingly presented in too simple a way for accuracy, then that is being simplistic. If one glosses over or omits relevant facts, that is being simplistic. And we're going to show that unfortunately that's what supposed evolutionary science is doing regarding the origin of life. What about the beginnings of life, cells, and genetics? The popular scenario for how life began on Earth begins with natural compounds believed to be present in the early Earth's oceans. These were acted on by different possible sources: hot springs, deep sea hydrothermal vents, volcanoes, earthquakes, and further interactions are also believed to have taken place: lightning, ultraviolet light, meteorites, and electric discharges from the sun. And all of those things somehow came together and poof, life began in the imagination of evolutionists. Now, on a plausibility scale, on the surface, that does sound plausible, like it could have happened. Back in the early 1950s, a scientific experiment was attempted to duplicate those conditions, and it has been misrepresented by the scientific community ever since. In 1952, doctors Harold Urey and Stanley Miller performed an experiment trying to mimic the conditions of the primordial soup in which life was supposed to have begun. Here's what they did. They had a heat source, and that produced water vapor. And into that water vapor was mixed methane, ammonia, and hydrogen. And they also passed through that water vapor a 60,000 volt spark. They had a cooler to condense some of the products that were produced in that, and a trap to remove them from the environment. And in the end, they produced trace amounts of 10 amino acids. And everybody declared that the experiment was proof that indeed that's how life began on Earth. Let's examine the experiment in greater depth. They had to separate the materials that were produced by the experiment from the very environment in which they were to prevent their destruction. Touted as a proof of life that could have spontaneously happened, the biggest problem is that this very environment they created was destructive to the amino acids they also created. Half of the molecules they made were toxic to life. And amino acids break down even in pure water, let alone in this primordial soup mixture that they tried to simulate. The other thing people don't seem to recognize is that there are over 500 amino acids in nature, but there are only about 20 that can be used to make proteins in living cells. 20 amino acids to make a single protein. And DNA is needed to make that protein also. And this experiment was performed before science even knew about DNA. You also need RNA to make DNA and to make the protein. Not only is this a huge leap of faith, it's a huge failure of logic. The presence of 10 amino acids does not equal the possibility of the formation of life spontaneously. It's like saying that you found a brick or even a dozen bricks and thinking that it's the same thing as having built a whole house. We can see that doesn't work. In any living cell, there are things called organelles, each made of specific kinds of proteins organized in specific ways to perform necessary specific functions within the cell. All cells have these parts: a cell wall, a capsule layer, a plasma membrane, cytoplasm or fluid within the cell, ribosomes to make proteins from the DNA plans, and of course the DNA plans themselves, with a minimum of about 250 genes. It takes 20 different amino acids in a specific order to make a single protein. It takes on average 3 to 4 million proteins to make even the simplest single-cell organism. The products of the experiment had to be removed to prevent the destruction. So this experiment actually proves that life could never have arisen naturally. In fact, in later years, Harold Urey, one of the creators of this experiment, said, "All of us that study the origin of life find that the more we look into it, the more we feel it is too complex to have evolved anywhere. We all believe as an article of faith that life evolved from dead matter on this planet. It's just that life's complexity is so great, it's hard for us to imagine that it did." So which is more credible? The Urey-Miller experiment shows spontaneous life possible, with toxins made and a trap needed to extract the results from the environment? Or the Urey-Miller experiment shows spontaneous life impossible, with toxins made and a trap needed to extract results from the environment? If you want to do a deep dive into the science of this, look up Dr. James Tour on YouTube. He's a very accomplished organic chemist and nanotechnologist, professor of chemistry, professor of materials science, and nanoengineering at Rice University in Houston, Texas, and a very outspoken critic of the false scientific claims regarding the beginning of life, because he knows where they're lying, and they won't debate him. So this is a typical animal cell. Proteins are made in all cells except red blood cells and a few others. To see this process of protein production, we begin in the cell nucleus, where the genetic information is kept. But we also need several organelles, such as the Golgi apparatus, endoplasmic reticulum, and ribosomes. All animals have their genetic information stored in chromosomes that are made up of tightly packaged bundles of DNA. DNA contains recipes for different proteins that are needed to make and maintain the various organelles and the cell itself. Each recipe we call a gene. Inside the nucleus, a specific gene or recipe for a protein is selected, then unwound and separated. In an RNA polymerase, a copy of that protein formula is made by creating a strand of messenger RNA. Once it is completed, the messenger RNA leaves the nucleus. The messenger RNA strand is guided to a ribosome, where the ribosome reads the recipe for the protein and from that recipe assembles a chain of specific amino acids in a specific order needed for that particular protein. The amino acid chain then moves into the rough endoplasmic reticulum, where it is folded into a specific shape needed for the protein in question. From there, the protein bundle is moved to the Golgi apparatus, where it is covered with the membrane and then transported to the place in the cell where it is needed. Kinesin protein tows the newly made protein along microtubules to this spot in the cell where it is needed. It does this by walking along the microtubules and pulling the newly made proteins behind it. There are dozens of these kinesin motor robots in each cell. All of that to make a single protein. And this first protein happened by chance? Remember, the Urey-Miller experiment did not make even a protein. It made trace amounts of about 10 amino acids, not all of which would have been the correct kind of amino acids for the making of a protein. Which is more credible? DNA tells ribosomes how to string many amino acids in a precise order, fold it into a precise shape, code it to be used in a cell by chance? Or DNA tells ribosomes how to string many amino acids in a precise order, fold it into a precise shape, code it to be used in a cell, all by design. Here are some additional facts. As we mentioned, 20 different amino acids are needed to make proteins. The minimum number of protein-producing genes a single-cell organism needs to survive and reproduce in the laboratory is somewhere between 265 and 350. There are that many different proteins that are going to be needed by that organism. It takes 265 to 350 genes minimum for a single-cell organism. 3 to 4 million proteins total, not all different, are needed to make a single-cell organism like bacteria. DNA is needed to make even one protein. And remember, the Urey-Miller experiment happened before DNA was even discovered. To make proteins, you also need these organelles: nucleus, ribosomes, endoplasmic reticulum, Golgi apparatus. And these first 3 to 4 million proteins and the 200 plus genes necessary to make them and any other organelles necessary for a living cell all happen by chance without any direction or design? Which is more credible? 3 to 4 million proteins needed for a single cell organism, and it also needs a nucleus, ribosomes, endoplasmic reticulum, Golgi apparatus, all happen by chance. Or 3 to 4 million proteins needed for a single cell organism, and also you need nucleus, ribosomes, endoplasmic reticulum, Golgi apparatus, happen by design. Jeff Tompkins, doctor in genetics of Clemson University, said, "That's the biggest problem with evolution, how life started, because you need DNA to make proteins, you need DNA to make RNA, and you need RNA to make proteins. So it's worse than what came first, the chicken or the egg." So which did come first, the chicken or the egg? Well, that's been an age-old question. On the evolution side, we have DNA, and we also need RNA. We need 3 to 4 million proteins, and we need a cell. And which one starts first? The Urey-Miller experiment was done before DNA was discovered, or anything about its intricacy. In 1953, Watson and Crick identified DNA as the master molecule that controls everything in living organisms. Their discovery opened a new world of unimaginable complexity of life. Let's look at that complexity. DNA carries genetic instructions for the development, the functioning, the growth, and reproduction of all known organisms and many viruses. DNA can be broken down into these constituent parts. Base pairs would correspond roughly to letters. Codons would correspond to words. Genes would correspond to paragraphs, and chromosomes would correspond to chapters. It averages about 1,000 base pairs per gene, and a minimum of 265 to 350 genes are necessary for the simplest life form. Not only that, DNA is a language. It cannot come from inanimate material. It had to be written by a mind, like all other languages we know of. Now, evolutionary theory is this, that life evolved from simple to complex. If that were the case, one would expect that the number of chromosome pairs necessary for various organisms would also increase as the complexity of the creature increased. But here's what we find. The Myrmeca ant has only one chromosome. A fruit fly has 4. A kangaroo has 8. A carrot has 9. A snail has 12. A starfish has 18. A raccoon has 19. A mouse has 20. A human has 23. A chimpanzee has 24. A skunk has 25. An elephant 28. A goat 30. A horse 32. An elk 34. Brown bear 37. Gray wolf 39. Sugarcane 40. Fern 80. Short-nosed sturgeon 186. If creatures evolved from simple to complex, this would not be the case. Darwinian evolution requires organisms add new genes as they evolve into more complex organisms. But there is no known genetic mechanism for adding a new gene anywhere in any creature. Which is more credible? DNA is a code language. Chromosomes don't progress in order. There's no way for DNA to add a new gene, but evolution happened anyway. Or DNA is a code language. Chromosomes don't progress in order. There's no known way for DNA to add a new gene. Evolution couldn't happen. Evolutionists say that earlier species evolved into new species by mutations. So mutations are the essential evolutionary mechanism. They create images like this, which can look convincing, but these images do not reflect reality. Mutations can't work as the mechanism of evolution. Not only are most of these disadvantageous, they are hardly ever passed on to offspring. One example people try to use to prove evolution is that creatures that get trapped in caves will lose their eyes and their offspring will be born without eyes and without normal pigmentation. These creatures haven't been in caves for millions of years. They sometimes are only there for 1 or 2 generations before these changes happen. They have adapted to their environment, but they have not become new creatures. When those creatures are taken out of caves, they regain their pigment quickly, and their offspring can have eyes again. So, summarizing the issues with mutations, most mutations are not beneficial. Most are not passed on to offspring. They generally don't add ability, they usually take away ability. Mutants stay the same creature, and mutations do not add complexity. So, which is more credible? Mutation, which generally takes away ability, is the major mechanism of evolution of species from simple to complex. Or, creatures may manifest mutations or variations, but remain the same creature they were created. Let's look at a simple genome from Mycoplasma genitalium. It has one chromosome, which contains 580,000 base pairs of DNA. That's 580,000 letters in its genetic code. Compared to E. coli bacteria, which has over 4.5 million base pairs. Or the human DNA, which has over 3 billion base pairs. As we mentioned, the smallest genome is a bacterium called Carcinella rudi, was taken the record for the smallest genome with just 159,662 letters or base pairs of DNA and 182 protein coding genes. So for this to happen in nature by accident, too many things have to happen, not just one after another over a long period of time, but they have to happen all at once. Which is more credible, the simplest genome of 159,662 precisely ordered base pairs to make proteins, organelles, and a living organism just happened by chance? Or the simplest genome of 159,662 precisely ordered base pairs to make proteins, organelles, a living organism happened by design? The DNA of a single human cell contains 3 billion letters. If it were represented in printed words, simply listing the first letter of each base word would require over 1.5 million pages of text. 2% or less of the genome instructs on how to make proteins. What does the rest do? Well, for years, this was claimed by evolutionists to be junk DNA. Richard Dawkins said, The simplest way to explain the surplus DNA is suppose that it is a parasite. That's from his book on the selfish gene. He also said the greater part, 95%, in the case of humans of the genome, might as well not be there for all the difference it makes. The rest of the DNA, apart from the 2% used for making proteins, was considered leftovers from different evolutionary stages. And then they created what was called the ENCODE project, and it was systematically mapping out the human genome. These data enabled us to assign biochemical functions for 80% of the genome, in particular beyond the well-studied protein coding regions. So not only have they found that the rest of the DNA does indeed have function, they had, by the time of this article, found 80% of it had a function. I have learned since that now it's up to 84. I expect they will find that indeed all of the rest of the DNA has a function and is not junk DNA left over from evolution like it was claimed for decades. What is the purpose of DNA? Well, it's how to make all the organelles in a cell. It regulates hundreds of chemical processes. It tells how to repair DNA. It tells how to make different kinds of cells and organs. And it tells how to make a whole body. Each part not only has to be made and in the right place, but has to perform its unique function. DNA tells the body how to fight various diseases through the immune system. This quote from Professor John Maddock, CEO of Genomics England: "The failure to recognize the implications of the non-coding DNA will go down as the biggest mistake in the history of molecular biology." So which is more credible? Since only 2% of the human genome is for making proteins, most of the rest is junk DNA, ascribed to evolutionary leftovers with no function now. Or, since 2003, the ENCODE project has already found the function of 80% of the human genome, including breakthroughs to understanding several diseases. In Darwin's day, this was about all that was known about a cell. There was a cell wall, there was liquid inside. At the center, there was something that was large, which we know to be the nucleus. I believe that if Darwin had known what we know now about complexity of a single cell, he never would have proposed his theory of evolution. Nuclei, which contain chromosomes, which in turn contain DNA, the endoplasmic reticulum, the Golgi apparatus, and the mitochondria are found in most plant, animal, and fungi cells. They have to all be there and functioning for the cell to be alive. Which takes more faith, to believe that this happened by random chance or that they were created by a creator? Place your parts in a box and shake it until you get a watch. The idea of evolution also supposes that all of those precise parts also came about by random chance with no direction or intelligence. Evolutionists will counter and say, but wait a minute, there are billions of years for all this to happen naturally. What they don't realize is that time is not evolution's friend, but its enemy. Amino acids can only last a few weeks in water before dissolving, let alone in the toxic environment that was created in the Urey-Miller experiment. They can't hang around for millions of years and wait for some mysterious process to randomly form a protein out of one of them, and then another mysterious process to randomly form 3 to 4 million proteins to make a living organism. And of the over 500 amino acids that we know exist in nature, only 20 are used to make proteins, and they have to be sequenced in a precise order for a particular protein needed for the cell wall or different proteins for various organelles that exist even in the simplest life forms. One single protein and 3 to 4 million needed for a cell. Time is not a magic wand. Even billions of years cannot make impossible things possible. Things that have never been observed, things that go against all known principles in science, things that are not even properly conceived, don't happen just because you give it lots of time. Which is more credible? 20 different amino acids must combine in only weeks in the precise order to make proteins and organelles into a living organism, all by chance. Or 20 different amino acids to combine in a precise order to make proteins and organelles into a living organism needs a designer. A living cell is an amazingly complex thing, and all parts of that system need to be present and working for the cell to live. It's impossible for this process to happen incrementally. You can't have half a nucleus or half a mitochondria or half a ribosome or half a Golgi apparatus and expect anything. This introduces a concept that you need to know. It's called irreducible complexity. The idea that all parts of a system are necessary for the thing to work. It cannot happen incrementally, as in the first part and then the second part until it is all completed. It just doesn't work that way. There are so many examples, even in the simplest life forms. For instance, many bacteria have what's called a flagella. That's a tail that moves them around and lets them get to food and evade predators. That flagella is powered by a flagellum motor. There are two different kinds, a proton motive force engine and a sodium ion pump engine. They're made up of from 23 to 40 different proteins. They can spin at up to 100,000 revolutions per minute, and they can propel a bacteria 60 body lengths per second of speed. Now, cheetahs can only do 25 body lengths per second. That would be the same as humans being able to run at 60 miles an hour. They can go forward and reverse, and all parts have to be there for it to function. These motors have parts like motors that humans make. They have the same function: gears, a rotor, axles, a drive shaft, bushings, a universal joint, and adapter rings. And remember, this is just one part of a single cell organism, supposedly the first kind of life to evolve. If you think that such things can come together strictly by chance, I submit you are not thinking logically. And the motors in bacteria are not the only kind of motor molecules that exist. We humans have a minimum of hundreds of thousands of machines like this in almost every cell in our bodies, and the bodies of all animals and plants too. The ATP synthase molecule, it's a spinning molecular motor. It converts food and air into energy for all cellular processes. It has 6 different parts. It spins at about 230 times a second, and there are hundreds of thousands minimum in each cell. Tens of thousands of ATP synthase molecules are in each mitochondria, and the average cell has 500 to 2,000 mitochondria. Liver cells have 1,000 to 2,000 mitochondria. Heart cells have 5,000 to 7,000 mitochondria. And human egg cells have 100,000 to 600,000 mitochondria. This is a perfect example of the definition of how simplistic the whole evolution scenario is. They are definitely presenting a subject too simply, misleadingly so, and they're glossing over or omitting relevant facts. They want us to ignore the unimaginable complexity of life and just wave their magic wands and say, well, life and evolution happened anyway. So which is more credible? Intercellular motors with 6 to 40 parts can't work without all the parts functioning, but this happened just by chance. Intercellular motors with 6 to 40 parts, which can't work without all the parts functioning, and this had to be created all at one time. Now, evolutionists have claimed for decades that 98% of our DNA matches the DNA of chimps. But this is only talking about a small portion of our DNA. It's not talking about the whole human genome. It ignores 98% of our DNA that is non-coding DNA. All animals would have similar proteins. So the fact that the 2% they were talking about would be that similar to chimps is not surprising. 98% of this 2% matches the DNA of chimps, encoding for proteins only. The rest of the DNA, instructions for building the body during gestation, creating each organ, fighting disease, DNA repair, and on and on and on. Hundreds of processes are regulated by the rest of that DNA. But even if their statement about 98% of the DNA of humans being similar to chimps, what they can't reconcile is that 92% then matches mouse DNA, 50% matches banana DNA, and 42% matches fruit fly DNA. So what does that prove? It's just a comparison of the portion of the genome that codes for proteins. This proves nothing favoring evolution. Which is more credible? 98% of 2% of the human DNA for protein coding matches chimp DNA. 92% matches mice, 50% bananas, 42% fruit flies, proving somehow evolution. Similarity of protein coding human DNA matching that of chimpanzees proves similarity of proteins only, ignoring the 98% of the human genome. Generally, we have DNA in two places in our cells. The first place, which houses most of our DNA, is in each cell nucleus, as we have mentioned. This is called nuclear DNA. Both our mother and our father, our grandmothers and grandfathers, as well as our great-grandmothers and great-grandfathers, contribute to our nuclear DNA. But in all of our cells, except our red blood cells, we have a separate bundle of DNA. This DNA gives instructions for the functioning of our mitochondria, which convert food into energy in each cell. Mitochondrial DNA comes only from our mothers and female ancestors. All humans alive today descend from one woman who has been dubbed by evolutionists as mitochondrial Eve. I would submit that all humans today do indeed descend from one woman, the biblical Eve, mitochondrial Eve. Calculated with evolutionary assumptions instead of real data, is supposed to have lived about 175,000 years ago. But calculated from actual data from living population, mitochondrial Eve lived 6,000 to 6,500 years ago. Which is more credible? All humans alive today descend from mitochondrial Eve. Calculated from evolutionary assumptions, lived 175,000 years ago. Or all humans alive today descend from mitochondrial Eve, calculated from actual populations, lived 6,000 to 6,500 years ago. Let's summarize what we discussed today. The primordial soup model is a guess that doesn't work, and it's not a viable explanation for how life could have begun. The Urey-Miller experiment really proves life could never have arisen naturally. DNA gives instructions for making each protein in several exacting steps. 20 different amino acids out of a possible 500 plus amino acids are used to make proteins. 3 to 4 million proteins of many different kinds are needed for a single cell organism. DNA and RNA are also needed to make even one protein, plus ribosomes, Golgi apparatus, etc. There is no known mechanism for adding genes to any genome, which is necessary for the evolutionary theory. Mutations usually subtract ability, not add ability, and they're typically not passed on to offspring. The simplest genome known has 159,662 base pairs in its DNA. That's impossible by chance. The junk DNA claim is false. 80% of the human genome already has been proven to have function. Given the fragile nature of amino acids, lots of time is the enemy of evolution. Irreducible complexity. All system parts are needed to work. It can't be incremental. 98% of the human genome matching chimps is only comparing protein creation. It ignores 98% of the genome. And mitochondrial DNA indicates that the first female ancestor lived 6,000 to 6,500 years ago. When you look at all of that data, you realize the idea of evolution is completely untenable. We come to this psychological principle called cognitive dissonance. When a person holds a core belief so strong that when they are presented with new evidence that goes against that belief, it is not accepted because protecting the core belief is so important. They will rationalize, ignore, or deny anything that challenges the core belief. It's not the creationists, but the evolutionists who have cognitive dissonance. They will not accept the scientific data. They are trapped in their religious beliefs of an origin from strictly material processes requiring billions of years and so many unfounded assumptions. So this is the fourth video in our series. We have looked already at the age of the solar system, the age of the Earth, the geologic record, and the origin of life. And we've seen that the data does not fit the evolutionary model, and it does fit the creation and flood model. Next time, we're going to discuss the fossil and coal record. I would encourage you to like, share, and subscribe to this channel. Thank you for joining me.